Hi to everybody. I will conclude this session. I'd like to come back on the aquatic environment and fish, especially, and we will talk about genetic diversity. The objective of my presentation today is to show that fragmentation can have an impact on fish population. And I will show you how we can use this genetic information for the cumulative effect of fragmentation. It is based on the works that we have done during these last years. I will start with something which is very basic and saying that fragmentation in aquatic environment is about obstacles, big dams, smaller dams, or wells. We know that it alters habitat and the movement of fish, and it also modifies the functioning of ecosystems and dynamics of populations and communities. I will give you a definition. It is about individuals interacting between themselves and the several habitats, and we have got ascending and descending movements. With fragmentation, we can have two cumulative effects. First, the reduction of the size of population, it is here, and alteration of movements between the different habitats. The populations constitute one of the first biotic level, so it is about impacts and identifying the potential of restoration. To study population in aquatic environment is quite complicated. It is expensive. We talked about pit tag. It is expensive, and most of the time we use indirect method. The one that I'm going to present today is about molecular level. So I represented the different habitats with letters. It can represent alleles or genes. So the fish, when they go from one habitat to another, they transport these genes and alleles. So we should find the same alleles in the different habitats. When we have got a fragmentation, it is the same principle. So a reduction of the population size, so it means that we have got less alleles in the population, and also a break of connectivity. So the population will be different from one side to another of the obstacle. We won't have the same alleles and genes. So we can use these letters to measure the indirect impact and also the evolutive health of population. Genetic diversity constitutes the adaptation capabilities of the populations to environment changes. In order to summarize, the genetic measure is an indicator of impact and viability of populations, and it is what I'm going to present you right now with this plan. First of all, a study showing how fragmentation can have an impact on genetic diversity and differentiation. A second study, I try to show you how we can use this genetic diversity in order to identify key parameters. You will understand later what we can do with this data and how to link genetic diversity to these impacts, and how to use genetic diversity to help to make decisions. I'll come back to my theory. So, theoretical impacts on genetic diversity. So we have got the same population. I've kept the alleles. We are in a non-fragmented system in this case, and we can see that we have got the same number of alleles, and it is the same identity in the two circles. So 2 habitats and a genetic differentiation that we can measure with FST. If we have got a fragmentation of the population, I've taken a first theoretical case with a partial fragmentation, and in the second case it is older, so alleles are disappearing. On average, the allelic wealth is going to be reduced, and if fragmentation is higher, the genetic diversity increases and the genetic differentiation is stronger. From a theoretical point of view, in fragmented areas, we have got a genetic diversity which is lower and a higher genetic differentiation. We implemented a study in 2 rivers in the south of France, the Célé, which is not really fragmented, with some mills, with fish passes, and another river which is much more fragmented, the Vior, with 2 hydroelectrical dams, downstream and upstream. So impact of the fragmentation, which should be higher. For each river, we have taken fish in 10 stations. That's what you have with the black dots. The fish that we decided to study are on the screen. We have got the common days, for example, Véron with 6 to 8 centimeters and Cheven up to 60 or 80 centimeters. So different sizes of the body of the fish. For each species, we have taken 25 individuals, we have taken the DNA, and we have studied that. And we obtained genetic data for each species, for each alleles on this graph. So for the fragmented river and the non-fragmented river in gray, you can observe for all the species a reduction of the allelic wealth. If we take species by species, we can see that for 3 of them, Cheven, Vendoise, and Goujon, well, the fragmentation is significant, and for the smaller species, we don't have a significant impact. So, generally speaking, it follows the theory, except for the smaller species. For the genetic fragmentation, we can see a higher differentiation on the black river. For the 2 biggest species, Chevet and Vendoise, the impact was higher on the fragmented river, and for the 2 smaller species, we didn't have a significant impact. Once again, we have got empirical data following the theory. An important impact then on genetic differentiation. As I have said, we are happy because our empirical data follow the theory. But with ground studies, we can have the hypothesis that the fact of the difference between the 2 rivers is not due to the recent fragmentation, but maybe due to older fragmentation. In order to go further on the causes, and it is the second part of my presentation, we have used the fact that DNA can capture recent and older history of populations. It is the same when you walk in the mud. Well, you can know if that person is small or tall or fat or thin. It is the same in this case. So recent or ancient history is not the same. It's what we try to use with this approach. Bayesian computation. I haven't translated it. The principle of this approach, it is to compare simulated data to empirical data of genetic diversity. So we have got knowledge, and then we implement scenarios that we imagine. For example, let's say that there was a barrier on a river 300 years ago, and on another river, no barrier. For each scenario, I formulate genetic data, and for each scenario, I'm looking for descriptive data. I repeat that 1,000 times with different parameters of size, migration, so that I cover all the possibilities. On the ground, you take empirical data. On this empirical data, we can have allelic differentiation, and you compare that to the simulations. And the idea is that the simulation with the smaller difference between empirical and simulated data constitutes the most probable scenario for what is happening in reality. So after this theoretical part, I'm going to show you the scenarios that we implemented. The first scenario, it is the double bottleneck. First of all, we've got the past at the top, and we imagine that we have got exchanges of populations. A first bottleneck due to effects that we don't know. So 2,000 or 4,000 years ago, this bottleneck reduced the population size. And then we had a more recent bottleneck, so maybe 300 years ago, with an hydroelectric dam. Here there is a fish missing. The second scenario, it is the same, except that we only have one bottleneck. The fragmentation that could have taken place due to recent dams have no influence on our population. The third scenario, it is the check scenario, considers that the population haven't changed and that they are steady in the time. So we realize simulations for each scenario and each river. And what we have shown is that for the Vior, it was a scenario of the double bottleneck. It was the most probable one. We have showed that with the first bottleneck, more or less 4,000 years ago, well, it is an estimate, and at that time, we had more or less 30,000 individuals. This bottleneck reduced a little bit the population. This population had a second bottleneck 200 or 300 years ago, and it reduced the population to 50 to 100 spawners. For the Vior, which is minimally fragmented, we have got a bottleneck maybe 3,000 years ago, and the population was similar to the one of the Vior. The reduction was due to the first bottleneck, and then after these individuals didn't knew another bottleneck. Thanks to that, we can quantify the major elements, and we can see the impact on genetic diversity. The differences can be due to historical impacts, but mainly due to recent effects and the adaptation, implementation of another bottleneck. The question of this reduction of genetic diversity, is it serious? Can it have an impact on fish, on their health and viability? In order to reply to this question, we looked at the common days on the Vior and Sélé, which follow them for 10 years now, and we have got demographical data. Here we have got population curves for the Vior, the highly fragmented river. We have got triangles and the arrow descending. What we can see in the Vior is that we have got a significant decrease of the common days. So we had only 2 or 3 individuals during the last years. And for the Sélé, in the same years, we can see a small increase of common days. In parallel, this population decline was accompanied on the Vior with a decrease of the number of alleles. So we started with a river with a higher allelic diversity, and with the recent population decline, it increases, so the differences between the fragmented and non-fragmented river. What we can see on the Vior is not due to fragmentation, but in the 1960s, we know that a parasite arrived and we had climate changes. The capacity of adaptation of this population are too weak, maybe, to face these new pressures on the Vior and on the Sélé. So we can potentially have consequences. So it can be linked to the demographic health of the population. To conclude my presentation, I'm going to show you one of our ongoing work. These data are to prioritize actions at the local scale. The objective was to deploy a method on genetic diversity in order to know which barriers are giving more trouble on a river. The idea with my small fish and alleles to work on the principle that more the fragmentation is high, and here you can see that on the drawing, more the effect is significant, more we will have an impact on genetic differentiation. So we can expect a small differentiation between the population, but if we have got big troubles with the whales, well, the genetic differentiation between upstreams and downstream will be higher. And this is this principle that we try to implement and to expand it on all the obstacles that we can find on a river. And we use that for 2 species with 2 indicators of differentiation, and in parallel for each obstacle we characterized these obstacles physically speaking in order to know which characteristics could explain these effects. So first of all on the Aru and the Sélé and Vior. So 12, 19 and 21 obstacles for each river with 2 species of fish upstream and downstream of each obstacles, and also the quantification of 4 parameters, 8 parameters in total. So I'm not going to show you all the results, but just with a graph. So we have taken the basic characteristics, height, slope, the fact that we have got fish passes, just to show you the result that we can obtain on a result. Here it is the Vior. The results were as good on other rivers. So the idea was to use a multi-varied method on the Héron and the other river. and to put them so each dot is an obstacle. And with this dimensional axis, we were able to calculate a crossing parameter. So here we have got a problem with the first one and the last one with a few problems for the crossing of the species. So the ranking, an objective ranking of these obstacles on this basis. So with the sampling, we had 50 obstacles more or less with different shapes. So old obstacles which were remodeled, and more recent wells in concrete. We realized that it was not this kind of characteristics that could explain the fact that the fish were crossing or not. But on the contrary, if we had a fish pass, we had genetic evidence of crossing in comparison when we had no pass. An important use of these passes for genetic diversity of population. To summarize and conclude, what I try to illustrate in my presentation is that DNA is a source of information on what happened 10 or 15,000 years ago, and also it gives us evidence of viability and resilience of population. So we can have information on impact and viability of populations. Moreover, the tools which are available and developed can give us fundamental information. It was my first part of my talk, and also for the management. This kind of analysis, well, we need to continue the development of analytical tools and to see which information we can get with this genetic differentiation, and also we need to analyze the tools that we can implement. I thank you for your attention.